what I will try to cover in the next 20 minutes, and I see my clock start running here, is some concepts about comparability. And also towards the end, I will touch in terms of how actually comparability itself is linked to the whole biosimilar development at this point, and what aspects of comparability can be applied towards biosimilarity and so on. And hopefully we can maybe have some questions towards the end of the presentation. Comparability has been one of the key evolving regulatory framework within the US FDA system and also on a global perspective. What I've highlighted here, in this, oops, sorry. Just trying to figure out the pointer is, nope, there's no pointer. What I'm trying to highlight here in this slide is some key milestones regarding the evolution of biologics regulation in the US, which are actually quite critical in terms of how the development of biotech products have evolved over these years. One of the key milestones that occurred very early on back in the mid-1980s was the points to consider that the FDA have developed, which is still actually quite a valid document that puts in place some concepts such as platform, generic validations, and so on. But it wasn't really until the mid-90s with the advancement of analytical technologies where the concept of a well-characterized biotech products came about. And the ability to be able to understand to a better extent how, what the structure aspects of the molecule led to the development of a concept of comparability where you could potentially make changes to your manufacturing process and so on without actually having to repeat the clinical requirements that are often needed for a new product registration. So back in the mid-90s, this concept started evolving that eventually led to the development of the ICH Q5E comparability document, which has remained to be one of the really major thanks for that. One of the major workhorse for the biotech industry that allowed that allowed actually the industry to introduce new technologies, to be able to introduce additional manufacturing sites, scale-ups, and so on, without having to perform clinical trials throughout all these changes. And where we are today, so after the comparability also, along the same time, the FDA also went ahead and replaced the previous PLA and ELA concept with the biologic license application, where also the manufacturing process, it's the product and the manufacturing site and the facility were not as intimately linked as before, where you can actually get a product license and you can actually manufacture a same product in multiple facilities. And where we are today, we are here essentially where we've been over the last several years dealing with the concepts of quality by design, and actually that has evolved quite rapidly in the US with the FDA pilot program, and I will actually show towards the end some of the approvals that have been obtained for both post-approval changes and also for new biologic license application under the QBD pilot program that actually has heavily utilized some of the comparability concepts. And if you look at comparability itself, when the comparability protocol concept was put in place back in the mid-90s, it was often applied to a single product and a single change. So in the early days of the practice of comparability, it was more or less limited to simple changes or restricted to one or two changes at a time. But then over the years, and with quite a lot of work through several scientific conferences, exchanges with regulators, the concepts of comparability starts being expanded, where as the regulators get more experience in terms of how to review packages, how to look at data and weigh in the risk-based assessments of the information related to comparability, what we have today, and actually this is one major development, the concept of an expanded change protocol or an expanded comparability protocol, where now it is possible actually under the FDA pilot program to bundle a variety of changes that can apply to multiple products or to multiple sites under the same comparability protocol, and be able to get these pre-approved in a way that will then facilitate significantly your supply network. And I will share with you towards the end an example of how we used these expanded change protocols to allow actually to apply to our entire biologic manufacturing network, to all our sites globally, and cover also all our products under one overarching protocol that gave us significant amount of regulatory flexibility, and also a downgrade of all the changes that you typically have to go through APAS to a CBE 30 or below in the US. And as I mentioned, a lot of these discussions regarding comparability and the well-characterized concept were first started back in 1996, where the first well-characterized biotech meeting that the FDA co-sponsored was put together in actually in San Francisco. And this event continues actually throughout the years, and it's now, we just had the 16th event again in San Francisco last year. This meeting also moved to Europe, where some of these concepts now are applicable with the element of post-approval lifecycle change management, which is essentially a version of a comparability protocol, but with a European terminology. And we're also glad to report that with a lot of work with JPMA and also with the support from PMDA, that we are actually taking the CMC forum now to Asia for the first time this year. And the conference that PMDA will host will happen actually in Tokyo with significant presence from regulators from the region. And one of the principal topics in that conference will be also on applicability of comparability and the advances in that regard. So moving quickly to what comparability is, and I think I'm going to go through this quite readily because we are all aware that comparability is a demonstration of high degree of similarity between products produced by either different manufacturing processes, different equipments, different sites, and so on. And it's really, an integral component of the life cycle management of a product. So I just showing here a schematic in terms of what a typical biotech product goes through throughout its development cycles, both in terms of the number of patients that they may be throughout clinical development, and also the scale that you may utilize throughout production. And definitely when you come to to commercial production, especially if you have big blockbusters or large volume products, you often have to manufacture these across the globe. So some of our products, such as Avastin and so on, is manufactured in 4 or 5 different facilities throughout the globe where we make the drug substance, and we have also multiple facilities where the drug product is also manufactured. But we are still in an environment where the ability to register multiple facilities is restricted in several countries, where you are only limited to one facility or so on. So comparability is really, in order to manage this type of changes which are essential to meet both the patient supply need, and often you make these changes not because they are internally driven. They are often the changes are driven by external changes in regulations. When we had the whole animal-derived raw materials scare, and we had to go back to all our old processes and try to remove some of the bovine-derived peptones and so on. This actually led to significant exercise of a comparability that actually is driven by external needs in order to meet future market demand. And over the years, we have done, I think, at Genentech and now within the Roche Group as a whole, probably over 40 or so major comparability exercises. So this is where we have a major upgrade to a process, or we have a addition of facilities for the various biologic products. And we always go back through that entire history of changes, and we do lessons learned in terms of what have we learned throughout the years in terms of doing this. And we often categorize our changes either under what we call a same process version or under a new process version, where we actually designate the material with a new process version that reflect probably a higher level of risk that is associated with the change. And clearly one of the biggest area of changes is site transfers. whether they are internally within the network or whether these are, or whether they are going to a contract manufacturers. This probably is account for roughly 60% of all our major comparability exercises throughout the network. And what we've also learned that if the site transfer is done without being associated with major process changes, so if you perform a site transfer but you tend to restrict your process changes to only facility and equipment fit, so the things that you need, because no two facilities are built exactly the same, so you have scale differences, you have equipment differences, but if you restrict the type of changes in the context of a site transfer to simply adapt to the new facility, we often find that these transfers tend to be low risk. And they're often being supported with the basic analytical comparability exercise. And rarely, actually, we never had to go to a point where we had to supplement a pure site transfer with additional either non-clinical or clinical data to support these type of exercises. But there's also the other type of changes where you actually introduce intended changes to your process. So that's where the cell culture is changes, or you change your purification process, or if you undergo change to your product configuration, such as formulation or new delivery. These type of changes tend to have a higher risk, and you find, based on the type of change, that you may actually have to escalate your comparability exercise and include additional work beyond the analytical or biological comparability. And so taking all that data into consideration over the years, we came up with this grading system that I think some of you may have seen before, where we categorize each comparability exercise with a letter, whether it's an A to E. And these represent the type of experimentation that's needed to support a given type of change. A tend to be the basic package, where it is predominantly an analytical, a biological, and a stability exercise. B is where you add extended biological characterization, and this tend to be for certain molecules that have, for example, FC effector function or some other type of biological activity in their mechanism that require a more broader biological characterization. C, actually, where we deem that the change is significant enough that we need to do a non-clinical experimentation. So tend to be an animal study. D is it where we actually perform a human PK/PD in support of the change, and E actually where we do an actual human clinical experimentation to support the change itself. And actually for a site change, for example, where you typically have no change in the characteristics, these are often, actually in all cases, they fall in category A, where they are simply supported by analytical and biological work. If you introduce cell culture or purification changes, and the cell culture changes tend to be higher risk than purification changes, you often find that you introduce some variation to the molecule that often exemplify itself through charge differences or some type of hydrophobic characteristics. So in cases like this, we may actually do an additional animal toxicology study in support of these changes based on the extent of differences that we do observe. And clearly, if you perform formulation changes, if you have new excipients, that often lead to having to repeat a human PK study. And in a case where we introduce a new cell line, which we haven't done yet for a commercial product, but which we do routinely in clinical development from the early stage to late stage, if we will have to introduce a new cell line in a commercial production, that definitely for us will go into a category E, where we will then do some type of a human clinical experimentation based also on the type of the change in the cell line, whether it's a new transfection or is a new host, and so on. So, but that's essentially where we categorize the type of changes for comparability itself. When comparability is performed, it's also very important to look at how you develop your acceptance criteria. So for our products, we don't just compare to the data that's been produced, from the last campaign or from a given site. We compile the history throughout all our productions. For some of our large monoclonals, we have now databases that include over 400 or 500, large-scale drug substance data, and all that information actually is always updated and collated, and the acceptance criteria is actually derived from that cumulative history of information. And that's how we then assess any future change against this complete data. And you also have to be very careful in looking at the profile. So often it's not just the numbers that tells you whether you pass your criteria or not. It's often the complex profiles. These tend to be often your ion exchange chromatography, hydrophobic interaction, depends on your type of molecule. But that's actually where you look at the overlay of the pre and post change, and you get a picture in terms of to what extent you see differences. And we actually do see differences. So when we make purposeful process changes in cell culture and so on, often you see differences in the profile. So it goes back to your knowledge in terms of, are these meaningful differences that you see here? And can you justify them based on your characterization data, based on the understanding of what these peaks represent? And these are actually real case studies in terms of some of our process changes over the years. So this is kind of where comparability has been done previously. What has happened over the last 2 years, which is really a major leap in the U.S. regulatory framework, is the introduction of the concept of an expanded change protocol. And the term expanded change protocol actually was the first utilized in a CMC forum in July, actually in Bethesda Hall at the NIH, and was coined by Steve Koslowski, who is now the director of the Office of Biotech Products. And it refers to the ability of bundling a variety of changes under a similar regulatory framework, and was perceived as a way to allow some application of QBD elements to biologics without actually having to change the CFR. Because if you had to change the CFR, this will go into essentially the next 10 years or so before you could actually be able to introduce these concepts. So the expanded change protocol was put as a mechanism by which one can leverage existing regulatory changes in the U.S. that's allowed by the regulatory system in the CFR, and see to what extent these can be further expanded. You probably are familiar with the Office of Biotech Products pilot program. But the pilot program, actually, it's almost towards its completion at this point. And within the Roche group, we have had 2 major applications in the pilot program. One of them is on a post-approval changes, which was on an expanded change protocol for site transfers. And that's what I mentioned earlier, where we bundled all our facilities under one overall protocol. And we had also another application. It's for a new biologic license application. Actually, it's currently under review. It's for pertuzumab, which it's an add-on therapy to Herceptin that most of you probably have seen the positive clinical data from that. And that's currently under review, where the review is done jointly by the FDA, EMEA, and we have several other agencies as observers throughout this process. It is the first full biologic license application under a QBD program with a full claim on a design space for the entire DS manufacturing process. So we're in a quite interesting observation mode to see how that will evolve. I can tell you from the first round of questions, there's quite some interest in that, and probably the amount of questions we received in the first round is beyond what we have received on all our previous BLAs combined. And that's after we had actually 8 meetings with the FDA throughout the pilot program. We had 4 meetings with the EMEA QBD or PAT team also, and we had also meetings with PMDA and also with various other agencies. So just to give you an example of how the expanded change protocol work, because eventually that will become the next evolution of comparability and will become practiced hopefully broadly also within the industry. Historically, we take, for example, one facility where we make a drug substance, we transfer it to another facility where we want to make the same drug substance. So what we wanted to envision is a collection of all our facilities across with all our products, and be able to have the ability at any point throughout our within our network to move a product A from any facility we have to any other facility, and be able to do all of this under a pre-approved mechanism. And with an agreement in terms of what information can be provided and how we can also get an inspection waiver, so that within the US that can be downgraded to a CBE 30. So we came with actually two expanded change protocols. The first one dealt with the facility itself. So this is the ability to convert a facility from a single product to a multi-product facility, from a single host to a multi-host, or to also combine clinical and commercial production in the same facility. So that was the first expanded change protocol that we got approved actually in first in the US, and in which we used risk-based mechanisms to actually weigh the type of experimentation that you will do and cleaning validation that supports a particular type of change that will then apply across the entire network. The second one, which was more unique, was driven for comparability. So it has at its core a comparability exercise to allow for product transfers throughout the network. And that's the one that was under the FDA pilot program, and actually was the first approval under this pilot program with the FDA. Each of these protocols have components in them. A scope of limitations that defines what can be included, what is excluded. Quality risk management, so that's the application of ICH Q9 extensively to how we go about performing such exercises. A comparability protocol embedded in the exercise, and then is a GMP component. It's something that we came up as a new assessment about inspection risk, where we will provide certain amount of information to the agency about the state of the facility in terms of inspection history, in terms of the GMPs and so on, that will allow then, and with a certain scoring, risk-based scoring, that if you meet certain criteria, essentially you fall under an inspection waiver. So that the regulatory submission itself can be categorized as CBE30. And for the ECP scope, so it applied to all our approved products across all our network, but it required that the facility that we are transferring to is licensed, because otherwise the inspection has to occur. It also required that when you do such a change, you are also limiting the amount of process changes that you do. So this will not be applicable to a time where we make significant process changes or we introduce a new cell line or so on, so that the risk associated with these type of transfers is still manageable. And this is regarding the multi-use ECP, which I think is more straightforward. So what we have used actually this combination of these two protocols to license our Singapore facility for biologic production, where we make 2 antibodies now in the facility there. And we were able to do so by I'll show here some of the timelines. So under the current PAS system and the current requirements, where you have to file also stability data, for example, for 3 months or 6 months, under this expanded change protocol, we also got a waiver from the FDA based on an extensive assessment of the stability profile of the products and the ability to correlate changes observed at elevated temperature to what we observe in real time. We were able to actually get the acceptance from the FDA that we file essentially with no real-time data. So essentially your submission can occur with the commitment that you carry the product on stability, but you don't need that stability to be included in the actual submission. You can only file with the accelerated stability data that we have shown through a lot of work initially that can actually correlate to what one will observe in the context of real time. So the combination of the downgrade from a PAS to a CBE30, along with the ability to file with no real stability data, allowed us to get our second antibody in that facility that we were able to release it to the US market almost 7 months in advance of what would have been a normal cycle. And now that same combination of this regulatory ECPs are being applied across all the network as we move products throughout all our facilities. And all future products that we file will actually be cross-referenced to this ECP. So our current BLA, for example, will have a cross-link to this ECP to allow all future products to also fall under the same categorization. So this is just an example in terms of how comparability have significantly changed. And I'm going to just conclude with this final slide, since we realize here we're in a biosimilar session, so I'm just going to say a couple words on that. As you know, comparability is done to confirm quality, safety, and efficacy. Similarity is done to establish quality, safety, and efficacy. So while what I have described here is not applicable from a regulatory perspective for a biosimilars application in the first time around, once a biosimilar is approved as a biosimilar, all the concepts of comparability will then be applicable to how then the biosimilar manufacturer will make subsequent changes to their own manufacturing process. So all of this eventually will be applicable once a biosimilar is licensed. So I think that has quite some applicability in terms of how to manage future manufacturing changes for both an innovator and a biosimilars manufacturer. Not the first time around, but after being initially licensed. And I think with that, I realize I just ran over a few minutes, so thanks for your attention.
Thank you very much, Wassim.